Dr. Joel Wertheim is an associate professor of medicine in the Division of Infectious Diseases in the School of Medicine here at UCSD. He conducts computational and bioinformatics research that helps to bridge the gap between evolutionary biology and public health action. He received his BA in biology from Wesleyan University, his PhD in ecology and evolutionary biology from the University of Arizona, and then undertook a 3-year postdoctoral fellowship at UCSD in our antiviral research center. During this time, his focus shifted from basic sciences on the molecular evolution and phylogenetics of RNA viruses towards translational science. Coronavirus, being an RNA virus, obviously was of considerable interest to him. He has studied the impact of HIV, the AIDS virus, through molecular surveillance in New York City, and has served for the past 5 years as a research consultant for the US CDC and for Centers for Disease Control and Prevention, developing protocols and strategies to improve molecular surveillance. He developed HIV Trace, a web application that enables rapid construction of HIV molecular transmission networks and facilitates near real-time HIV molecular surveillance. That is, keeping track of transmission within an epidemic by means of very careful examination of the genetics of the viruses. More recently, he has studied the timing and origins and spread of the SARS-CoV-2 pandemic. The combination of bioinformatics, epidemiological modeling, and phylogenetics that he has been developing and employing is revolutionary in virology and public health responses to epidemic infectious diseases like HIV and the series of COVID epidemics, Ebola and MERS virus that has now culminated in COVID. Joel has been interested in coronaviruses well before the current epidemic. In 2013, he published a paper on their ancient origins. For several reasons, the origins of SARS-19 that is causing this epidemic has remained uncertain and controversial. Joel has contributed to the understanding of this question by applying phylogenetic methods to the dating of the earliest cases in China. I look forward to hearing his current thoughts on the origins and other issues related to the COVID epidemic.
Excellent. Alan, thank you for that lovely introduction and for inviting me here today. I just want to make sure that I am on spotlight for everyone.
Yes.
Everyone's seeing me big? Great. All right. So I'm Joel Wertheim, Associate Professor of Medicine, and had to teach remotely during the COVID pandemic. So I picked up a couple of Zoom tricks that I hope to show off today, in addition to walking you through what I see as the most likely case for the origin of SARS-CoV-2, the causative agent of the COVID-19. In this talk, I'll be going over a study where we wrote a review looking at the early origins and also some of our original scientific work led by my graduate student in the biomedical informatics program here at UC San Diego. All right. So if we want to know where if we want to understand the origins of the SARS-CoV-2 pandemic, we have to start asking some very general questions. First one might be, where? Where did it start? Well, most likely in China. And here to orient you to the country, we have a map of China. Right here in sort of the middle, you can see Hubei province and the city of Wuhan, a city of 11 million people, home to the Wuhan Institute of Virology and several live animal markets. And this is where we first saw the first cases of SARS-CoV-2 at the end of 2019. But as you may know, the viruses that are most closely related to SARS-CoV-2, which are found in bats and circulate widely around the world, but the closest related bats found all the way over here in China, southern China, in a region called Yunnan province. And here they found several closely related bat viruses, including some whose name apparently occasionally appears in the news media, like RATG13. Keep in mind that one for later. Now, what's notable here is the considerable amount of land between Yunnan, where these closely related bat viruses are found, and Wuhan, which led a lot of people in the beginning of the question to ask, how did a bat virus from southern China make it all the way to Wuhan, urban center of 11 million people? Well, before we get too carried away with this question, I'd like to turn the clock back to 2002 and 2003 with the SARS pandemic, the original SARS epidemic, which had outbreak here in Guangdong and in Hong Kong, which, as you can see, is also a considerable distance from Yunnan, even though the closest bat viruses to the original SARS virus were also found in Yunnan. Interestingly, and often forgotten, is that there were also SARS-1 viruses found in Hubei province at the time of the original SARS pandemic. So it should be no surprise to anyone that coronaviruses can move from Yunnan around China, whether through bats themselves or potentially through intermediate hosts often used as part of the fur or meat trade. But about a year ago, the WHO was very interested in trying to figure out where SARS-CoV-2 came from. So they dispatched a crack team of scientists to Wuhan to investigate. And their report, which I'll refer to throughout this talk as the WHO report, was published in early 2021 and concluded that the source was likely in Wuhan, most likely from an animal intermediate host, not to spoil the ending. But one thing they did note was that the first cases identified in the second week of December 2019 tended to be associated with animal markets. Not all of them, as you can see here in orange, but quite a few of these earliest cases were associated with these animal markets in Wuhan, suggesting that this may be a place to investigate further. Now, although I don't have formal training in epidemiology, I like to fancy myself a weekend epidemiologist. And that makes me think, I want to figure out where a pathogen came from. I should go all the way back to the early days of epidemiology and a fellow named John Snow, who figured out that a water pump was responsible for the cholera epidemic in the mid-1800s in London. He famously removed the handle from the pump, possibly ending the epidemic. But he was able to do this by mapping all of the cases of cholera shown here around that pump and realized that that pump was the central location and likely the cause of the cholera outbreak, even though then they didn't even know what cholera was. Still hadn't fully adopted the germ theory of disease. These spatial analyses, where we look to see where diseases are in space and time, can be quite informative. And if we go back to Wuhan, here we have an aerial view from Google Maps. We focus in on this building right here. This is the Wuhan Wholesale Seafood, the Hunan Wholesale Seafood Market located in Wuhan, China. It is actually two buildings separated by a large road, and contrary to popular reporting early in the pandemic, these are most definitely indoor buildings with roofs and walls. And if we focus on the cases inside of this market, and we look at the stalls at the market, we can see that one of our earliest cases reported by the WHO happens in the western part of the market, shown here. That's in December 13th. Then when we get to December 20th, we see a whole bunch more cases on the western half of the market. And it continues to spread out from there as December continues until December 31st, which was the last day that the market was open and operational, or it was closed by the authorities. So if we apply our Jon Snow type thinking here, we can see that there seems to be spread within the market over time and from a particular part of the market, this western section of the market, just west of that road I showed you. What's notable is that the rumors of live animal sales were said to be conducted in the western building and less so on the eastern side of the market. So that's our first spatial signal that the market might be important more specifically. But then if we fan out and just look at, say, earliest cases in Wuhan at the end of December 2019, where did they live? You can see there's a lot of clustering right above the river there at the Huanan seafood market. We don't see a whole lot of clustering by the WIV, or the Wuhan Institute of Virology, where some of the most eminent coronavirus researchers in the world have their labs. And as we look at the spread around this market over time, we can see here we have beginning of December, we get to later December, and now we get to the end of December, and we can see the spread radiating out from that central point in the market, strongly suggesting that that is the geographic location where SARS-CoV-2 first started spreading in people. All right, I think we can generally agree on where. Spread likely started at or around the market, but now the question is how. Now, there are a lot of ideas about how this virus first got into humans, but I really would like to turn it over to the eminent John Stewart to explain he thinks the coronavirus first got into humans.
There's a chance that this was created in a lab. There's an investigation.
A chance?
Well, but
oh my
if
God.
there was evidence, I'd love to hear it.
There's a novel respiratory coronavirus overtaking Wuhan, China. What do we do? Oh, you know who we could ask? The Wuhan Novel Respiratory Coronavirus Lab. The disease is the same name as the lab. That's just a little too weird.
Okay, so fair point. There is a eminent coronavirus research lab in Wuhan, which at the beginning of the pandemic, a lot of people thought was very geographically suspicious. But I think this is where we turn it over to Stephen to talk some sense into him.
I'll tell you what I'm suspicious of. I'm suspicious of the Daytona Beach Spring Break Herpes Lab. That might be where all the herpes comes from.
All right. Makes a good point. You want to study coronaviruses where they jump into people, although I do have to quibble with Stephen a bit because I happen to be an expert in where herpes viruses come from. And here, if you read this news article, it says we got genital herpes from making sweet love to chimpanzees. It's with no shame that I acknowledge that this is an actual news article written about an actual paper I wrote in 2014 about the origin of herpes virus in humans. I would love to go into the details here. Rest assured, this was not a conclusion from our paper. But yes, you would study viruses where they are found naturally. But that said, John isn't entirely wrong here. Viruses do leak out of labs, and it's been well documented. There was a SARS outbreak due to a lab accident in Singapore that was well described in 2003. We've even had pandemics that have been restarted by viruses that were previously in labs, specifically the 1977 Russian flu, which was an H1N1 virus that spread around the world unexpectedly. If we look here at the genetic relatedness of all of these different flu viruses, we can see that the branch of this tree, which indicates all of the genetic diversity of the viruses that's leading to the 1977 pandemic. That branch extends over 20 years of time. But if we look to see how many genetic changes lead up to that branch, there are none. There's only one place I know of where a virus can sit for 20 years without changing one bit, or 27 years without changing, and that's in a freezer. And it's very likely that the 1977 influenza pandemic virus had been in a freezer for at least 27 years before it reemerged. But that said, this was a well-known and well-studied virus and likely escaped during a vaccine challenge trial. And we had known about that virus for at least 50 years before it escaped. That said, we have seen at least one novel human virus that had never before been known to science escape from a lab. And that was the Marburg virus, which first started infecting people in Europe and in Marburg, Germany and Yugoslavia due to contaminated monkey or infected monkeys and infected monkey cells. However, what's very important about the Marburg virus is that when they did contact tracing on these first cases, shown here, all of the cases of Marburg could be linked back to workers in these biotech labs or their spouses. So the epidemiology was very clear that Marburg came from a lab because we can trace it back. Unlike for SARS-CoV-2, where when you apply those same epidemiological approaches, you end up back at the market. So speaking of that market, shown here, when the WHO issued their report, they were very emphatic. They said that there were no live mammals being sold at the market. They said there was seafood, both live and killed, and there were animal products, but that there were no live animals like, say, civet cats or raccoon dogs, which act as intermediate hosts for SARS, or something like mink, which we know can get infected on fur farms in Europe. People who had been shopping at the market for years swore to the investigators that they had never seen a live animal sold at the market. Well, that didn't sit very well with a colleague of mine, Eddie Holmes. He's an eminent researcher down in Sydney, Australia. And he had previously been to that market in 2014, taken there by the Chinese CDC as an example of a place where coronavirus spillover could occur. And he took these pictures. This first one shown here just shows the regular day inside of the market. Not particularly clean, but as we look at this, his next picture that he took in 2014 doesn't quite agree with what the WHO was told. Because if you look here, you can see on that bottom cage, you have a bunch of birds, but up top, those look very much like mammals. And in fact, they're not just any mammal. Those are raccoon dogs, which are bred. They're wild animals, but bred for fur and for meat. And here we have photographic evidence of them being sold at the Hunan Seafood Market 6 years before the pandemic got started. Now, what's remarkable here is that the location of this cage where Dr. Holmes took this picture is in the southwestern part of the market, exactly where we see cases starting to emanate from and positive samples from the environment in the market. So sort of suggests that the WHO report wasn't complete. However, shortly after the WHO report was published, truly damning evidence came out in the journal Scientific Reports. Now, this was a group of Chinese and European and American researchers who had been tracking the sale of animals and in markets, Wuhan wet markets, I mean, they sell live animals around Wuhan for years leading up to the COVID-19 pandemic. In fact, they had been documenting viruses that live on ticks infecting these animals. And they documented, both with photographic evidence and just documentation, a whole host of different animals being sold there. Now, again, I'd like to you to focus. You can see hedgehogs, you can see snakes, you can see what look like to be guinea pigs, and in the lower left-hand corner here, if you look closely, that is a raccoon dog. This is an animal. Zoom in here. This is an animal that has been experimentally shown not only to be able to be infected with SARS-CoV-2, but also to be able to transmit it via aerosols to other raccoon dogs. So this animal is an ideal intermediate host that this group of international researchers documented was being sold at the Hunan seafood market leading up to the start of the pandemic at the end of 2020. So I got to tell you, I'm a little suspicious about what the WHO was told. Just doesn't add up. Kind of looks like a smoking gun to me. All right. So I think we've established where, and we have a pretty good idea of how. What about when? Well, when is where my research really comes into, because that's the question that I've been asking myself since the start of the pandemic. When does this thing start? At the beginning of the pandemic, we're all wondering, maybe it's been around for years and we're already immune. That wasn't the case, but it was wishful thinking back then. The WHO, they did a pretty good job trying to find all of the earliest cases they could come across. And essentially, although this document predates the WHO report, and it's actually what we knew at in the early 2020, that there were cases in the first and second week of December, definitely in the second week of December, and they started to increase. And many of these early cases were linked to the market. And then as the pandemic took off into January, we got more and more and more of these early cases. So that kind of suggests the pandemic taking off in late 2019. Of course, remember, for the original variant of SARS-CoV-2, it took several days before anyone would even become infectious, maybe a week before they were sick, and a couple of weeks before they would be hospitalized. So, and since most of these early cases are based on hospitalization or severe symptoms, we have to think that the virus should have been circulating for a bit longer. Also, keep in mind, even for those earlier variants, a lot of people who got SARS-CoV-2, even with no pre-existing immunity, had completely asymptomatic infections. We could have missed those first people who got infected, and they may not even known that they were sick or just had a mild, unremarkable cold. Okay, but you started to see these other reports from around the world, like this one, published in the Science of the Total Environment, suggesting that there was SARS-CoV-2 in human sewage in Brazil going back as far as November 2019. We also saw some claims that sewage in as early as March of 2019. I'm not going to dignify that paper here. Then we also have these claims of early cases in Milan, Italy, in the same region of Italy that got hit the hardest during the first wave of the COVID pandemic, way back in early 2020. And here, there's claims that in early December, a 4-year-old boy, PCR positive for COVID-19, meaning it had RNA from the virus in his body. Now, this boy, a 4-year-old, went to a measles clinic with symptoms dating back to November 21st. He had no travel history and no known contact with anyone else who had been sick. But they were able to amplify SARS-CoV-2 genetic material from his sample. Now, what's weird is that they weren't able to do it the way you and I would go in for a COVID test. They had to amplify it. Nothing came up. And then they went and they amplified the whole thing again. Now, for those of you who, like me, haven't set foot in a wet lab in at least a decade and maybe never, if you just have a single copy of SARS-CoV-2, say from a positive control, to make sure that your diagnostic assay is working. And just a single copy of that viral genetic material finds its way into your testing sample, you could very easily amplify a positive result using this method. So keep that in mind. All right. And then we have this paper that was put out by some researchers at Harvard at the end of 2020, where they said, based on satellite imagery of Wuhan in fall of 2019, that the hospitals there were overwhelmed with parked cars, indicating that they were full up of COVID patients in September and October of 2019, long before anyone had acknowledged the pandemic had existed. So, is it possible? So when did SARS-CoV-2 first jump into humans? Well, when I answer that question, I look here. This is how I see SARS-CoV-2. 29,000 A's, U's, B's, and C's. Nucleotide bases, which are color-coded here. And anytime you can see a faint variation in color going from green to blue in a single row, that indicates a mutation within the SARS-CoV-2 gene. And this is how I see the virus. Thousands and thousands of strings of nucleotides. And we look for the changes in these nucleotides, understand which viruses are more closely related to each other, and how fast they've been evolving. So if we look at the virus from the very, very beginning of the pandemic, we can see that it falls into what we call two main lineages. Had we been interested in it back then, we could have called them variants, but they didn't get variant names. They're just lineage A and lineage B. Lineage B, as you can see here, has a lot more early samples in it, those colored in really dark purple from the very end of December. We don't really see a lot of lineage A until January. There's one sample from December, but the rest comes about in mid to late January. And every point on this tree is a virus that was sampled. And those branches or lines leading to the point indicate that separating those different viruses is a mutation. You have either 1, 2, or 3 mutations that separate each virus. Now, although we have some very fancy and complicated bioinformatics algorithms to build these trees, you can see here in the very, very early days, there wasn't a lot of genetic diversity, and you could essentially look to see, okay, this virus has these 2 mutations, which it shares them with another virus, they're closely related. Really nothing very fancy going on here. But you can see that in the beginning, there wasn't a whole lot of diversity except for these 2 major lineages, A, B. And B seems to have been sampled first. But if we want to know when they occurred, we can't just ask when they were sampled. We have to turn to our old friend here, the molecular clock. The molecular clock is a tool that I've used throughout my research and actually dates back almost 70 years in concept. But has been very powerful in understanding the spread of infectious agents, especially SARS-CoV-2. So what is the molecular clock? Well, it's just a simple observation that the number of mutations that a viral genome picks up over time is relatively constant, which means which means that you can look to see how many mutations does a virus have, and that can give you an idea of when it would have been sampled in time. You give me any SARS-CoV-2 virus, I can just look at how many mutations it is from the very base of the tree, and I could probably tell you within a month or 2 when that virus was sampled. That's how good this clock ticks in the virus. As every month passes, more mutations accumulate. And we see that in the actual SARS-CoV-2 data. There are currently over 6 million SARS-CoV-2 genomes that have been sequenced and made available to researchers. This is just a sample of them, but if we plot all of those genomes by the date that they were swabbed, and we count the number of mutations they have from the root, you can see it's this very clear linear progression. And on average, SARS-CoV-2 will pick up about 24 mutations, substitutions, from the ancestor of all of SARS-CoV-2 every year. So, and so using that, we can estimate at what point in time would the ancestor of all SARS-CoV-2 have existed. And we did that using the data set I showed you earlier. And then we can get an estimate of the genetic ancestor of all of SARS-CoV-2, that virus that gave rise every SARS-CoV-2 genome that has ever been sampled in the world. And we can tell you with reasonable confidence that it happened sometime between mid-November, mid-December. That's when the genetic ancestor of SARS-CoV-2 existed. Pretty cool, but not actually the whole story. Because, and this is I actually published like the cocktail napkin, that you can see here the genetic ancestor, or what they call the time of most recent common ancestor, or TMRCA, is that dot all the way to the right. That could actually happen after the date of zoonosis. Now, why is that? Because you could have a zoonotic event and then some people whose transmission chains go extinct. And if those transmission chains go extinct, they won't be represented in all of the genetic diversity that we see worldwide. And so we would miss that early window between when the first person got infected and when the genetic ancestor. Now, this is kind of a complicated topic, so I've prepared, well, an analogy that I think might help. So, regardless of your field of study, you probably have heard of this woman, the mitochondrial Eve. Now, she didn't live in the Garden of Eden exactly. She likely lived in East Africa. And she lived 155,000 years ago. And this woman is all of our genetic ancestor. As a matter of fact, every human being, their mitochondria, that little organelle genome that lives inside of all your cells, is all descended from this one woman who lived 155,000 years ago. Now, humans as a species are a lot older than 155,000 years. And there wasn't just one woman that lived 155,000 years ago. But back then, mitochondrial Eve, who is shown here, colored in dark black, well, every year, every generation, when she was alive, her and all of the other women reproduced. They gave rise to descendants, and some of those descendants survived, and some didn't. Then every generation, a different proportion of surviving descendants would exist. Certain mitochondrial lineages would be lost, and as it progresses forward in time to present day, all that is left in the world are descendants of her. And every other mitochondrial lineage that was present at her time has gone extinct. So there we all are in the bottom row, 155,000 years later, and we all trace back our ancestry to this one woman, at least in terms of our mitochondria. And you can think of the same thing happening in SARS-CoV-2, where you can have a zoonotic event, and then you have all of the genetic diversity. But the question is, does that date of zoonosis, is that the same as the genetic ancestor, which you can see here, where at the time of zoonosis, all of genetic diversity that is ever going to be is birthed? Or would you possibly have a whole bunch of stuttering transmission chains, where you have the zoonosis indicated at the star, and then slow and sort of limping transmission, where like one or two people get infected, but they don't lead to sustained transmission until at some point an explosion of transmission. And that's where we get our genetic ancestor. That's where lineages A and B and all of their diversity come about. So our mitochondrial Eve, or our SARS-CoV-2 Eve, happens a long time after, weeks, maybe months, after the zoonotic event. We don't know which one is the case. Oh, this is where we figure it out. All right, so we know what the genetic ancestor is. We have to find a way to link them to that primary case, that first person to get infected with SARS-CoV-2, that date of zoonosis. And how can we do that? Well, the good news is I work at UC San Diego, and one of my former students, I sat on his committee, a guy named Nima Moshiri, he's now teaching faculty in computer science. And back when he was a graduate student here, he published a program called Phabites, which essentially simulates large viral epidemics. Now, he and I worked on this study and published it before SARS-CoV-2 was even a thing. But we realized that his package would be crucial in bridging this gap and figuring out how long did it take to go from the date of zoonosis to all of the genetic diversity that we see. Now, to do that, you first have to employ a very complicated epidemic model, which I'm going to make big just because putting up a slide like this makes me seem sophisticated. To be fair, it's kind of over my head. But this is an epidemic model where people can become susceptible. They start off being susceptible to infection, then they become infected, then they go through a pre-symptomatic period where then they can transmit even though they don't yet have symptoms, and then they can be ascertained and diagnosed. They can end up in the hospital, or they can be recovered. And this is basically a mathematical model to express all of the early spread in Wuhan, China. It was actually developed specifically to model the Wuhan epidemic starting in January. We just extended this model a bit back in time into December and asked, here is our workflow, which, while technical, only has two real important parts to it. The first part is we use the molecular clock, ask, when was the SARS-CoV-2 Eve? When was the genetic ancestor of all of SARS-CoV-2? And then we ask a second question, how much cryptic transmission could have happened before Eve? And to do that, we use our epidemic model to say, how long can you transmit before you have to give rise to the genetic ancestor of all of SARS-CoV-2? So we combined both of these models using Bayesian phylogenetic inference, which sounds a lot more sophisticated than it is. And we got our best estimate of timing for a SARS-CoV-2 spillover, shown here, where we put our best estimate that SARS-CoV-2 first got into humans in either likely in late November 2019, possibly going back into October, although certainly not in September. And here we have what is essentially the first estimate of that primary case of SARS-CoV. We can do questions now or at the end.
I think it's probably best to wait till the end.
Yeah,
all right, we'll get to the end. Okay, so let's okay, so our 4-year-old boy in Italy, do we think there was international transmission to infect a 4-year-old in Italy in late November? Well, based on the number of cases that we would estimate in Wuhan, China at the time, no, there probably would have been fewer than 20 cases infected in mid-November. Very likely any of them could have gotten on a plane and infected anyone in Italy. So
nope.
All right. What about this claim of overwhelmed hospitals in September and October 2019 in Wuhan? Well, if we look at the hospitalization numbers, Now, there's no way to get a massive uptick in hospitalizations till at least mid to late December. So,
Nope.
thank you, Lana. She's very helpful in answering these questions. All right. What about the intelligence report that 3 Wuhan Institute of Virology staff members became sick and hospitalized with a respiratory pathogen in November of 2019? I should mention was at the height of a really bad flu season in China. Well, do we think we could have had enough hospitalizations in all of Wuhan by mid-November to get 3 people who worked together hospitalized? Lana?
Nope.
I didn't think so either. All right. What about some work done in the US population? It was a seroprevalence study by the CDC where they looked at around 7,000 people and looked for SARS-CoV-2 reactive antibodies in January of 2020, before the pandemic took off here. And they found that 84 of their samples were seroreactive, meaning that they had antibody response to SARS-CoV-2 in January of 2020. And they said, well, that means it was probably here. But that means 1.1% of their population is seroreactive, 7,000 people. Now, we do a little bit of math, and we say, population of the United States around 350 million people, 1.1% seroprevalence rate. If you think about that study, that would say 3.6 million people were infected with SARS-CoV-2 in the US by January 2020, and we didn't have any hospitalizations or a spike in deaths or illness? I'm sorry, that bunk. Don't buy it. All right. Now, there have been some other discussions about the early days, accusations that our estimates of the early pandemic are unreliable because Chinese researchers obscured genome sequences that were put online and then were deleted to mask the origins of SARS-CoV-2 and mislead researchers. This was based on a paper published by John Cohen in Science Magazine, who lives up in Cardiff. Well, do we think that this is true? Well, the claim is that we see a lot more lineage B viruses in the early days, especially linked to the market. But lineage A viruses, and this is kind of a subtle point, are actually more similar to viruses circulating in bats. Now, they're more similar by only 2 or 3 mutations, but that led a lot of researchers to say, well, lineage A viruses must have come first, and then lineage B viruses evolves from them because lineage A looks like bats. And if lineage A came first, why don't we see any lineage A genomes at the market or linked or even in December? Well, the Chinese must have deleted them and hidden their existence. Well, that's an idea. I'm going to give you all a little bit of preview of a paper that we just getting ready to submit, and hopefully will be out in the next couple of weeks. So please don't report on this, but we think there were actually 2 separate spillovers of SARS-CoV-2 at the market. One that led to lineage A and one that led to lineage B. So even though the A virus actually happens to be closer related to bat virus, if both of those viruses are found in an intermediate host animal in the Wuhan market, and the B virus jumps over into humans a week before than the A virus, which the data suggest it did, then you could get more B virus than A virus at the beginning of the pandemic. A could still be more closely related to bats, and it would explain how there actually aren't any hidden or deleted bat virus, human viruses out there, but in fact, we would just have 2 spillovers. And that I think is most clearly shown here, where we have a bat virus that jumps into an animal in the market. You have a bunch of diversity within that market, and then the one virus jumps in late November and the other in early December. And that would give rise not only to the genetic diversity we see, but also why we didn't see any A viruses at the market, because E had already been growing exponentially for maybe up to a week. All right, now, 2 spillovers? Is that real? Is that right? I mean, that would take a lot of misfortunes to even lead to 1 zoonosis. I mean, think of everything that has to go wrong to get a zoonotic virus that causes a pandemic. First, you need a successful bat virus. The virus has to bind to human ACE2 cells. It has to have furin cleavage. Then it has to infect a secondary host, like a raccoon dog. Then it has to be able to transmit within that secondary host. Then it has to be exposed to humans. Then it has to be able to jump into humans. It has to be able to replicate in humans. And then it has to be able to transmit between humans. You need to get through all those 9 things just to get 1 pandemic.
But that will never happen. 3 misfortunes, that's possible. 7 misfortunes, there's an outside chance. But 9 misfortunes, I'd like to see that.
I admit, very unlikely. And there's only one man we can turn to in our time of need to explain this to him. And it, George Clooney. Just seems like he's going to be helpful here. All right, George, what do you got?
Right. Who are you looking for?
Tenzing Norgay.
Tenzing Norgay.
Right. Who's Tenzing Norgay? Is he like a worker at the market? I don't understand. Wait, is he the primary case, the first person to get infected with SARS-CoV-2? He's patient 0?
I doubt it. Tenzing Norgay was the Sherpa that helped Edmund Hillary climb Mount Everest.
I don't get it. Oh, okay. This is why Tenzing Norgay is so important, understanding multi-spillover. Because for 155,000 years, since mitochondrial Eve, human existence, not a single human being ever climbed Mount Everest. Then, on May 29, 1953, 2 men summited Mount Everest on the same day. 155,000 years and it had never happened. And then it happened twice in a single day? Well, this is what we call conditional probability. You see, once you have a bad virus, that can bind to human receptors and has furin cleavage and can infect a secondary host and can transmit a secondary host and can be exposed to humans and can jump into humans once and is known to replicate in humans and is known to transmit in humans. If all of those things are already in place, that same virus jumping into humans again, knowing that there's exposure, well, that's not reinventing summiting Mount Everest. That's having everything else in place and just following the leader. Tenzing Norgay. All right, now, actually, this is quite common when it comes to coronaviruses. When you look at the original SARS epidemic, that jumped into humans on multiple occasions from a much more genetically diverse reservoir. If we look at MERS in camels, that's repeatedly jumping into humans again and again and again and can't even sustain transmission in humans. It goes extinct so quickly. But because the MERS virus can jump into humans and transmit among humans, all it takes is the proximity to camels, have it happen again and again and again. All right, so that brings us to why.
Is that why? That's a real question, isn't it? Why? The how and the who is just scenery for the public. Oswald, Ruby, Cuba, the mafia. Keeps them guessing like some kind of parlor game, prevents them from asking the most important question, why?
All right, admit that's a totally different conspiracy theory, but a lot of similar thinking going on here, because there are a lot of conspiracy theories about why SARS-CoV-2 got into humans.
If I was to guess, this virus started transmitting somewhere in September, October in Wuhan.
September, October.
That's my own view. It's an only opinion. I'm allowed to have opinions now. You know, I am of the point of view that I still think the most likely etiology of this pathogen in Wuhan was from a laboratory. escaped. Other people don't believe that. That's fine. Science will eventually figure it out.
No offense to Dr. Redfield, but I think science has figured out, and it wasn't September or October, it seemed to have a natural origin on our hands. But that interview definitely didn't help things move forward, especially when you think about that bat virus, RATG13, which certain researchers, particularly this guy, Jeremy Lantham, has suggested was isolated from a bunch of miners in the Mojang mine in Yunnan province, where that virus was first isolated. And there were 3 miners who were killed by some pathogen of unknown etiology in 2012. And he has made the suggestion that they actually got RATG13 out of those miners and manipulated it into SARS-CoV-2. Now, I am not a medical doctor, but some medical doctors went and they looked at all of the symptoms and signs in those miners who died. And here they've just highlighted in a couple of them all of these signs and symptoms that don't match SARS-CoV-2, COVID-19 at all, like mucus and other such blood clots. And all of those signs and symptoms they claim are far more consistent with fungal infection. So those miners probably didn't die of a coronavirus, probably a fungal pathogen. Also, this is an important point about evolution. If the RATG virus is here on the tree, evolution, if you want to say manipulate a virus, you go forward in time. You don't go backwards in time, deep into the evolutionary diversity, 50 years back in time, undo 50 years of evolution, and then re-evolve it past other bat viruses that you didn't know about. That's just not how evolution works. So RATG13, if anyone mentions it to you, just remind them, it's a red herring. It's just a related bat virus. It's not the origin. It doesn't speak to the origin at all. Also, since the discovery of RATG13, other researchers have found a whole bunch of viruses that are even more closely related in Laos, which is right next to Yunnan, China. So there are a lot of bat viruses down there in southern China closely related to SARS-CoV-2. Don't fall for the RATG13 trick.
I do not believe this somehow came from a bat to a human. And at that moment in time, the virus that came to the human became one of the most infectious viruses that we know in humanity for human-to-human transmission. Normally, when a pathogen goes from a zoonotic to human, it takes a while for it to figure out how to become more and more efficient in human-to-human transmission.
All right. 2 things. First, SARS-CoV-2 is not the most infectious pathogen in human history. We got things like smallpox and measles. They are very, very, very transmissible. Part of what made SARS-CoV-2 so nefarious to spread on the world is that it had an asymptomatic period of infection, which means people could transmit without knowing they were infected or ever infected. Also, this idea that it had to be manufactured in a lab because otherwise it would take a while to spread. Well, if SARS-CoV-2 wasn't good at spreading at the get-go, it wouldn't have spread. Lastly, the claim that this virus was eminently adapted to spread at the moment it arrived in humans, and it's just not true. It was definitely good enough to spread around the world, but shortly after it got into humans, the virus picked up this mutation, E614G, also known as Doug by those who study him. That is the virus that took over Italy and New York and all of North America and Europe. It had an adaptation that allowed it to dominate the world, and lineage A, which had previously been circulating, fell by the wayside anywhere it encountered D614G. Now, what about, but the virus didn't stop evolving there. As you remember, about a year ago, heard about the Alpha variant circulating in Kent, the UK, and then that swept around the world along with some other variants. And Alpha was definitely more transmissible than the variants that had come before it. And then, of course, who can forget the lovely Delta variant, which became more virulent and more deadly. And of course, let's not forget the reason for the season, why I'm talking to you from home and you're all remote as well, the Omicron variant, which is unbelievably transmissible. This is what viral adaptation looks like. This is why we have more cases today than we've had in the entirety of the pandemic, because this virus is figuring us out. It just started off doing pretty good, but man, is it worse or more infectious. So this idea that SARS-CoV-2 is ready to go in humans, and that could have only happened if it were created in a lab.
Nope.
There she is. Yeah, just, it's not true. Okay. So now I'm going to get to the real reason why. And the reason why SARS-CoV-2 was so infectious is because of these guys right here, the dinosaurs, which we don't have anymore, except for the small ones with feathers that fly around, because the rest of these, your Tyrannosaurus Rex, they all went extinct around 65 million years ago. They're gone. Extinction is crucial to understanding why SARS-CoV-2 was so effective at spreading from the get-go, why it was able to cause a pandemic. When we do our simulated experiments and we let just a single spillover event happen into humans, only 70% of them go extinct off the bat. They infect one, maybe two people. Only 30% of them manage to transmit enough to establish in our simulation enough to essentially cause a pandemic. That's a virus that is as transmissible as SARS-CoV-2, still goes extinct 70% of the time because it just doesn't, because your average person with the virus doesn't transmit. Now that's in a highly connected urban area. If you were to do this same simulation in, say, a rural area where people have fewer contacts, 95% of simulated epidemics in rural areas go extinct. Which means even if you have a virus capable of causing a pandemic, unless it happens to establish itself through sheer dumb luck on behalf of the virus, you'll never see it. So in order to get two spillovers, we probably needed to have at least 11 spillover events for two of them to be successful. And I'd like to bring in to mind an analogy here to end. So why was SARS-CoV-2 successful and unique? Because SARS-CoV-2 sits here, way above the waterline. Highly infectious virus that once it gets started is guaranteed to continue to transmit among humans. Other viruses like SARS, when that jumped into humans, it took a lot of public health effort to bring it down. Otherwise, it would have continued to spread at a much, much higher mortality rate. Other coronaviruses like MERS, well, they sit just below the waterline. MERS is constantly jumping into humans, but it can never get that foothold because it's just not transmissible in people, which means it just keeps going extinct and extinct every time it jumps in. Way down here, those are all of the epidemics and pandemics we never see. Those are viruses that just aren't capable of infecting humans, just aren't able to sustain transmission. So SARS-CoV-2 isn't unique any more than it's just the end, it's just tail end of the distribution of infectiousness. It's just the virus that sits high enough above the waterline that can spread around the world. And with that, I'd like to thank all of the co-authors from the previous studies I showed earlier. And these gentlemen, especially Jonathan Pekar, my graduate student in the upper left, Nemo Shiri in the upper right, who is faculty, and then Conrad Scheffler and Mike Warby, who are both instrumental in guiding that project. And with that, happy to take any and all questions you may have. Thank you.